So friends, today I am going to tell you about the microscopy. The word microscope is made up of two words, micro plus scope. Micro means small things, tiny things, and scope means to observe, to see. So microscope. Actually, Anthony von Leeuwenhoek is considered as father of microscopy, and actually first compound microscope was built by Hans and Jensen. They were son and father's duo, and they were lens makers, spectacle makers. They made lenses and fixed two lenses together and observed the things larger than normal, which our eyes can observe. So Hans and Jensen in 1585 they made first microscopes. So they are considered as discoverer or inventor of microscope. So friends, the first microscope or lens which we can say, the journey of microscope started with the formation of lenses only, specs only. So this is the simplest kind of where we can enlarge a lens, right? Where we can enlarge the object, right? And you all know about that. I think almost all the houses we have this, in all schools we have this kind of this is also called as magnifying glass. So that is having one lens only, single lens. So in other words, we can say this is the simplest kind of microscope, right? Where an object can be enlarged little bit at certain extent. That here we can also say as we can magnify. I'll explain you later on what is magnification in microscopy. So it is part of optical instruments. Optics means where light is useful, something we can observe by use of light, and where lenses are involved. So this is known as, there are various types of microscopes. Simple microscope and compound microscopes are there. Various types of compound microscopes are also there, like phase contrast microscopy, fluorescence microscopy. We will discuss one by one about all these things. But the journey of microscopy started with the simple type of microscope. Why it is called as simple microscope? Because it is having only one lens, right? That is called as eyepiece. That is called as eyepiece, right? And here you can see it is mentioned as 20x, right? So a small kind of eyepiece, and no other objective lenses are there, only eyepiece, one lens is there. So when we are using one lens to observe, to magnify, to enlarge the objects, then it is called as simple type of microscope. See here, simple type of microscope, only the natural light we can use, right? And light used to fall on the mirror, and the light reflect and it pass through the, this is called as stage, this is called as mirror, this is stage, and over the stage we have to put our slide. Over the slide we have to put our specimen which we want to observe under the microscope. So simple type of microscope, right? Highly adjustable, right? Now in these days these are present as specimens only in the old laboratories, old institutes, right? Nobody is using that, but highly useful things when we were doing our experiments, especially for to see, to observe flower parts, stamens, even ovules, these kind of things, even pollen grains also we can observe through that, right? So a simple kind of microscope example is this only. This is the base of the microscope, this is the arm of the microscope, and this is called as knob for the adjustment, right? Adjustable knob, it can adjust based on the distance of the specimen from the lens. So wherever it is, we find that it is clear. So base, this is the adjustable knob here. Only one type of knob is present in simple type of microscopes, that is called as chorus only. There is no fine knob here present. And this is the stand, and with the stand the arm is containing one eyepiece. So this is called as simple microscope, where only one lens we are using to observe the small specimens, right? Friends, this is an example of compound microscope. Why it is called as compound? Because two types of lenses we can use, two lenses we can use actually here, right? This is called as eyepiece. This is called as eyepiece, that is having one lens here. Eyepiece, that is one lens, and the power of here it is lens is 10x here, right? And this is present on one tube, that is called as lens tube, right? So our eye, we have to put our eye here to observe the specimen. So that is why it is also called as eyepiece, right? We can change it. Now we have 10x, even we can change it with the 20x also, right? It enhances the resolution and the power of magnification also, right? This is the tube, lens tube. This is the body where all these lenses and things are attached, and this is called as the nose of objectives, nose of objectives. So these lenses are called as objective lens. This is eyepiece. Another lenses are attached with the objective lenses, right? These are replaceable. We can replace these, right? These are replaceable. We can replace these, right? So one lens is present here, that, right? And these are also of various powers, right? It starts from 10x. So 10x this, 10x this. So this, then it is 100x. Same way here, I think this is 20x, right? Even till 40x, right? 100x we can put on this nose. So it is called as nose piece for the objective lenses, right? So this is objective lens. With that, we can see here this part of the microscope is called as stage to put the slide, right? And to hold the slide, we have clamps here. These two steel things are called as clamps. Clamps used to hold the slide like this, right? So that slide won't move, specimen won't move while observing, right? Down you can see here, this is called as condenser, which allow the light, how much light should pass through the specimen. So this is called as condenser. Condenser is adjustable. So how much light is needed for better resolution, better magnification, better resolution basically, so that we can observe the specimen, right? This is called as mirror again here also. So here also natural light only we can use. We have to adjust it. We have to adjust it so that light will fall on that, and then the reflected light will pass through this, through the path, and in between somewhere specimen is also there, that is thin section only. So thin section only can be observed. The ultra thin sections we can observe better, right? This is the arm of microscope, and this is adjustable. Here two types of knobs are there, right? Upper knob is called as chorus adjustment knob. So chorus or adjustment, large adjustments we can do here, right? And the another one is called as fine adjustment knob, right? If you can see here, another part is here, right? Part of this only. This is the nozzle piece where objective lenses can be fixed. This is eyepiece here. This is another type of compound microscope only. The difference is here, you can see here binocular, right? Two eyepieces are there. These are also replaceable only. We can change it, right? And based on our requirement we can adjust. Many things are adjustable here, right? And even based on our eyes also we can adjust like this also, right? So distance of in between two eyes we can make it. It can be a trinocular, another space is provided here, right? This we can open it, and this space is for the fixing of camera. So camera can be fixed here, right? And we can take photographs, whatever the things are present in our specimen. That kind of facility is available here. This is the arm only, right? This is the stage. As we saw in the compound microscope, simple type of compound microscope, in that we saw this is the stage. These are the adjustable clips. right? A slide can be adjusted here like this only, it, right? And here facility is there to adjustment of these slides also, it can move like this. What is important here, it is connected with the wire here, electric circuit here, and if it is connected, then we can switch on and use it. Yeah, so this is the back portion of this microscope. It's made up of Lebomed only, which this microscope we are using from last almost 10 years and working fantastic in our laboratory, on my laboratory only. So here you can see now this is the source of light, so there is no mirror here needed, because source of light is LED light only, natural light we are not using, and it is giving better type of magnification, and a clear resolution used to be there, so better images we can get, right? And this also we can adjust, one adjustment knob is there, right? How much light is needed based on our requirement and based on our specimen, what specimen we are observing under the microscope. So this is LED light, source of the light, and you can see here this is here again a highly sophisticated adjustable condenser, right? And we can control how much light is needed, right? Some extra facilities are present here, right? Stage is also very sophisticated, right? And these are the two knobs are present here, right? One knob for the movement of slide, entire stage used to move, right, with the slide, so we can move slide and stage, right, in to and fro, left and right, and another opposite direction also. No need to, once we fix the slide in the clamps, no need to touch the slide again, right? By use of these knobs we can to and fro in direction we can right. Even the condenser and the position of light also is, one knob is available, we can fix that, right? How much light is needed, accordingly we can fix that also, right? Then this is the base of that microscope, base of the microscope, arm of the microscope. Here highly sophisticated type of knobs are present, two knobs are present, like in compound microscope we saw two separate knobs for coarse adjustment knob and fine adjustment knob, right? Here also the same thing are present here together. The outer knob, right, that is for the coarse adjustment. Coarse adjustment means it is adjusting the stage with the objective lens, right? You can see that movement, you can observe here, the stage is moving up, right? So that wherever the better resolution, better clarity is there, where we can fix it, right? So, and then when we reach certain stage that now specimen is observable, we can see that through the eyepiece, then we can, for fine adjustments, right, we have to use this fine type of knob, fine adjustment knob. So this is also a compound microscope only, but trinocular, binoculars are here, third can be used here, that is for the camera only, and source of light is also there. So adjustment of stage also it is there, so no need to touch slide after that. So this is our slide, on that we can put a specimen, right? This is the adjustment of light, now we can switch it off, right? So friends, little bit more sophisticated instrument in this series, that is also a type of compound microscope only, right? This if you we are using this from last 4 years, and the brand is a very good brand, right? It's German make, and costly instruments, so highly sophisticated things are present, so we have to use carefully. So but fantastic results we are getting, right? Cells, nucleus, even chromosomes we can observe here, and on daily routine basis we are using this microscope. Here you can see these are the binoculars, right? We can adjust based on our eyes only, right? And that is having one very good camera, right? So whatever the things we are we are observing on the slide, we can capture the photographs here, and this is connected with the we can connect it with the system, either laptop or desktop system. So whatever the things are we are observing on the microscope on the of specimen, right, these can be captured directly. These images or videos we can make and capture and store in our system, because facilities, USB is there connected with this, right. So this is extra facility. A good microscope is having always good camera is needed, so that images we can capture, we can make videos also. These can be stored in even hard disk, right. Here we can see 2-3 extra facilities are present. This is called as phase contrast microscope. As well as with compound microscope, we have here fluorescence facilities also there. So it is also called as fluorescent microscope, phase contrast microscope, right. Things are almost, basic things are same only, advanced things are different, right. These are eyepieces. Then this is the revolving objective nose, right. We can right, we start with the lower power first, then higher power, and finally this is having 100x, right. When most of the things are with better clarity and magnification, with high resolution we can, right. And based on our requirement we can adjust here also, right. Various kinds of filters are present because phase contrast now, right. And this source of light is also inbuilt here, and light used to adjusted here also, right, how much light is needed. So adjustment knob is also there, right. And with that, this is stage, clips are also there, the movement of stage is also there. Slide we can move with the stage, no need to touch once we install it. These are the same coarse and fine types of adjustable knobs. With help of that we can adjust the specimen based on distance from the objective lens, and wherever better clarity is there we can find out. Friends, this extra facility is here, right. These are the filters, different kinds of filters present here. So what kind of light is needed, that particular filter we can put and observe under the microscope, right. And the last facility that is available with this is this one, right, and that is part of fluorescence microscopy. So extra facilities are available with that. So this is all about some kinds of microscopes, right. Even one more microscope I will show you, just wait for some time. Okay. So friends, as I told you, other types of microscope, this is called as stereo microscope. Here the things are just opposite actually. These are the eyepieces, right. Another lens is present here, right, and the specimen we can put here, that will be visible here with the eyepieces, right. This is camera of stereo microscope. Stereo means it can give us a 3D image of any kind of specimen and no need of even section also. So thin sections are not needed, right. Basically people who are working in metallic field, metals, they used to observe through this. We used to observe somatic embryos and whatever the things are present on the surface, 3D view of that we can get by use of stereo microscope. So a specimen we have to put here at this place, and then that image will be captured here, and we can observe that image through the eyepieces, right. We have to observe here only, right. Much adjustments are not there, but a good stereo microscope we have, that is made of Olympus basically. And I think this microscope is almost 15 years old, we are using in our laboratory. One kind of adjustment we have here, that is the adjustment with the stage only, directly entire part of the all the lenses used to, right, assembly used to come up and down. After that fine adjustment we have to do, right, with the in between eyepieces and the objective lens, right, for better clarity we can find, right. So fine and coarse. Here the this is the source of light here, actually LED light, right. So light used to fall on the specimen, and the light reflected from the specimen used to send to the lenses, objective lens, then the eyepieces, right. So that image used to created in the eyepieces, which we can observe here. So source of light is here now. In other microscopes, compound microscope, we have seen source of light is back only, from the down only, back only. Here the source of light is from the up only, right? So that as much as good clarity of the specimen is there, right? The same thing we can observe through the eyepieces here, and the images can be captured by use of camera also, and that is also having a facility of USB. It can be connected with the system. So light quantity also can be adjusted by use of another knob, that is light quantity can be adjusted based on the requirement. We can adjust it. So these are some of the very old microscopes we have. Still these are in working condition. See here, right? I think more than 40 years old microscopes are there. Same eyepieces and nose for objectives, movable, right? And 10x, 40x, 100x type of objectives we are putting here. This is the stage. Movement is also still it is in working condition. This is the source of light, then condenser, then the aperture to pass the light. Another microscope, Lebomet, very old, I think more than 25 years old. This is a very old type of camera, still it is in function. Eyepieces and nozzle and objective lenses, source of light, condenser is here, and the stage aperture for the supply of light. Very old, older than that, right? Again, right? And here this is trinocular, right? This is again one more compound microscope that we have put one another facility, that is called as camera lucida. Those students who are in colleges, they may understand, they may heard about this word camera lucida, right? What is important here, by use of camera lucida, whatever things we are observing here in our specimen, side by we can draw the diagram of that also. That facility is camera lucida is providing. With that we can measure the size of the specimens also, even we can measure the thickness of cell wall, all these things. So micrometry is also part of this only. So camera lucida attached with the compound microscope, right? This is called as camera lucida. One day one video I will make for camera lucida and micrometry alone only. So this is for your basic information, right? Thank you.